He's like, when's the last time you turned it off? I'm like, I don't turn it off, it's a phone.
Yeah, you should.
And then I think to myself, it's not a phone, it's a computer. And I should remember from the beginning days of computers that whenever your computer is whacking out on you, you just do a hard reset. As a matter of fact, most computers back in the old days, they had a little kill switch, a little red switch on the side of the box that you just flipped on and flipped off. It was the breaker for the whole computer. I'm like, duh. So I turned it off, turned it back on, everything was good.
Isn't it the worst when there's like an easy fix to things, but you've taken so
Well, yeah,
long to try
I
to
just don't
find
normally
it?
think of a computer as fitting inside of your back pocket. My first computer that I had when I was in grad school, it had a 210 megabyte hard drive on it. It was the largest hard drive you could buy for a personal computer at the time. It was roughly the size of a brick, like a building brick, 210 megabytes. I walk around with like 512 gigs hanging around my neck on a flash drive. It's crazy what has happened. Okay, so for Monday, you asked what is next. Monday, read chapter 2 in Poland. This will start to talk about a little bit about economics, but what I want you to pay attention to is I want you to pay attention to Fritz Haber and the invention of nitrogen fixation. It's all in there, but read the whole chapter, it's a quick read. This is industrial nitrogen fixation. Fritz Haber won the Nobel Prize in Chemistry for this invention because this invention transformed agriculture in fundamental ways. He did this back in the late 1800s, 1879, I think, was the date that he first discovered this. So when we do start getting into history, which we'll get in, so this is going to serve as our introduction into nutrient cycling. We'll spend a couple of days on nutrient cycling. And then once we finish nutrient cycling, we'll be done with science to a large degree for the semester. And we're going to be talking about economics, and we're going to be talking about history. And our history will begin in roughly 1879. We'll go back periodically and talk about the early 1800s because we have records on, for example, the amount of the American populace that was involved in farming back in the early 1800s. It was much greater than it is today. But our deep coverage of history will begin in 1879, and it will go up to the Nixon administration. Because during the Nixon administration, they changed fundamentally the way agriculture gets funded by the federal government, and it's done since then in a way that was very different than the way agriculture got funded after the Great Depression and the New Deal. So we'll cover history and then economics and a lot of other stuff as we go. But that's where we're headed. Remember that October 4th is your group reports on the National Academy of Sciences GMO report. And the other thing that I want you to see is that I have also put a key to exam 1 up on the site. It's in the week that we took the exam, so September 15th to September 21st, but there it is for your review. So we finished up last time talking about genetics per se, and I began to introduce the idea of genetic engineering, which we're going to get in today. But one of the things that we talked about last time that we talked about it because we looked at the evolution of corn oil production and how if you select for higher corn oil production, you'll get higher corn oil production. But there's this cool visualization that was done by Harvard University. It's a thing called the Harvard megaplate. And they have a video of the Harvard megaplate on YouTube. And that thing that we saw with corn oil, that basically was just evolution happening. This process by which, if you have a heritable trait, you subject it to some sort of selection, then you're going to see an evolutionary response to selection. I gave you this. This is the breeder's equation. And so if we know how strong selection is acting on a population, we know the heritability of the trait that's being acted on, we can predict what's going to happen to that trait in the future. So the Harvard megaplague is just an example of this using bacteria. The nice thing about bacteria is that they grow quickly, and I'll just let the people from Harvard explain it to you. So this is available on YouTube. All you have to do is type in Harvard megaplague, and it will pop up. Thanks. And we cannot get sound for some reason. Last time I couldn't get the sound turned down. This time I can't get sound at all. Okay. Okay, it says it should be functioning. This thing seems to not be working. Oh, it's all the way up. Interesting. I did play something the other day on this, didn't I? Nothing. Okay. Let's try this.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up,
All right, let's go back.
so what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, porous thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So this idea that evolution occurs is not anything that shocking if you've ever heard of anybody being affected negatively by antibiotic-resistant bacteria. You guys who are athletes might occasionally have to deal with MRSA, methicillin-resistant Staphylococcus aureus. It's a antibiotic-resistant bacterium that you occasionally find down in the Navy Center. And when you do find it, they have to close down the locker rooms, and they clean the place with bleach, and then they open the locker rooms back up. I don't know, a couple of times a year it seems like they have to shut down for something along this nature. So that was over the course of 11 days. You went from bacteria that were susceptible to antibiotics to bacteria that were really antibiotic resistant over the course of 11 days, because they started out without any challenge, then they gave them a little challenge, then you gave them a much bigger challenge, and then a much bigger challenge, and then a huge challenge. And what happened was there's variation in the population of bacteria, and when they get to one of those boundaries where there are now more antibiotics in the agar, those individuals that have a variation that allow them to withstand that level of antibiotics, those are the ones that make it into that next band of antibiotic containing agar, and they're capable of growing there. And all of the strains that were not capable of doing that just died at that boundary. Remember, you could see where the boundaries were even after all the bacteria had populated the plates. And so this can happen relatively quickly. So this equation is very good at predicting what's going to happen in the future with these different populations. In chapter 2, in chapter 3 of Poland, he talks about antibiotic-resistant bacteria and also bacteria that are resistant to the stomach acids of cattle. And so when we get to chapter 3 and we start talking about how we raise cattle in the modern agricultural system, this idea of bacteria being resistant to environmental challenges is going to come back up. So I wanted you to see that because basically what we finished up talking about last time was essentially evolution. So we were talking about genetic engineering. Last time we had introduced how you do this using bacteria, and I was about to start talking to you about CRISPR because we listened to the CRISPR story about the woman who had sickle cell anemia, and they basically were attempting to cure her of sickle cell anemia by basically killing all of her stem cells that produce adult blood type and then upregulating or turning back on the gene that produces fetal blood cells so that she can just live on fetal blood cells that don't express this, this mutation. Do you have any questions over the first part of the genetic engineering stuff? Okay, then let's begin to talk about CRISPR. So CRISPR is a relatively new thing, but it's kind of taken the biological community by storm in a sense. CRISPR was discovered, it's another bacterial system that biologists have essentially co-opted to do their bidding. But basically CRISPR is a system by which you have an RNA molecule here, and this RNA molecule, remember that RNA has complementary base pairing with DNA, because this is how we actually make messenger RNA. Remember from the central dogma, central dogma is the term that biologists use to describe how you take DNA, you transfer transcribe this DNA into messenger RNA, and then you use that messenger RNA template to translate the genetic code out of the language of nucleic acids into the language of amino acid sequence, which basically make up proteins. And so this first process of transcribing messenger RNA, remember, involves the complementary base pairing of DNA nucleotides, which are things like adenine, thymine, guanine, and cytosine, and RNA nucleotides, complementary base pairs such that adenine combines base pairs with uracil, thymine base pairs with adenine, cytosine, and guanine. So there's a different type of RNA nucleotide, a different type of nitrogenous base that is used in messenger RNA. But because RNA can do this complementary base pairing, you can basically create an RNA sequence that will recognize a specific part of the genome of an organism. And it will go there and it will attach itself to the DNA molecule. So this molecule here is called guide RNA because what it does is it basically recognizes a section of DNA, opens up that DNA, and allows this messenger RNA this RNA molecule to essentially complementary base pair with the DNA that is there. And so in this way, the guide RNA targets a very specific place in the genome. So you can make this guide RNA be whatever sequence of RNA that you want, and in that way you can control where the CRISPR-Cas9 system is going to function. And then you combine this guide RNA molecule with what's called the Cas9 molecule, which is in the next slide. And the Cas9 molecule is this big protein, and it's capable of cutting DNA. And so it's giving you like little Swiss Army knives here with a little scissor attachment as kind of your visual for it. But basically it allows, this protein essentially allows a researcher to go in and cut either strand of DNA. It can cut both strands of DNA, it can cut one of them leaving the other one intact. And so it makes it capable to cut open DNA. And when you cut open DNA, you can do a couple of things with it. You can just cut it, and then once it's cut, it no longer functions because the genetic code has been disrupted. The other thing, though, that you can do with it once you've cut it is when you've cut it open, just like with restriction enzymes, when we use these with the bacterium for getting the plasmid to insert a gene into a plant's genome, once you cut it, you can also insert new pieces of DNA. And so in this case, you have your guide RNA combined with Cas9, and becomes a complex. And then once that complex is together, this guide RNA inserts itself into the DNA molecule that is there. And in this case, it's just basically being used to cut the DNA that is being targeted. And in this way, you can essentially destroy the function of a gene by basically targeting where you want CRISPR-Cas9 to act and then cutting the DNA at that point. Once the DNA is cut, that linear sequence of nucleotides, linear sequence of nitrogenous bases is broken, and then that gene no longer functions. And so the simplest thing to do with CRISPR-Cas9 is to single out a specific gene and essentially turn it off by cutting it. But this isn't the only thing that you can do with CRISPR-Cas9. You can also attach molecules to the Cas9 molecule. And we didn't talk in great detail about the structure of a gene. We didn't talk about the structure of a gene at all. But remember that a gene is a sequence of nitrogenous bases. These are very long, in some cases thousands of base pairs long. And so if this represents a DNA molecule, I'm not being careful to match these up, but there are adenines, thymines, cytosines, guanines, etc., all down the length of this. Well, the gene begins at a particular place, and it will be transcribed this way when you go to make a messenger RNA molecule. And this beginning of the gene is where the gene starts transcription. Upstream of the gene, sometimes thousands of bases, upstream of that place where transcription begins, there are going to be sections of DNA that are capable of binding with other molecules. And what happens is these places upstream of the actual gene, the name has just escaped me. This happens occasionally. My brain goes off track. Oh, these molecules that bind here are called transcription factors. And what these transcription factors do is they bind to the DNA, and then they bind to another molecule, a big molecule called mediator. And the mediator basically gets these transcription factors in contact with this part at the beginning of the gene, and it's these transcription factors that effectively turn the gene on and tell it to start transcribing a messenger RNA molecule. And so these transcription factors control whether a gene gets turned on or turned off. And so what you can do with the CRISPR-Cas9 is you can basically put on this Cas9 molecule essentially transcription factors, some of which will turn that gene on, or some of which will turn that gene off. So rather than taking the DNA and cutting it, what it does is it basically goes upstream of the gene that you're interested in regulating, and you either have a repressor, which shuts off expression of that gene, or you have an activator that increases the activation of that gene. And so with CRISPR-Cas9, you can basically just destroy the function of a gene, or you can regulate the gene, and you can upregulate it so that it makes more of the gene product, or you can downregulate such that it makes less of the gene product. And so in this way, you can have a lot of control over the action of specific genes, because the guide RNA tells you where exactly in the genome you want to do something, and then the Cas9 protein allows you to do a wide variety of things to that gene. In the case of the woman with sickle cell anemia, she went in, so in the case with sickle cell anemia, she went in, and you remember that they talked about giving her chemotherapy. Normally, chemotherapy is used when you have cancer, and it's used to kill the cancerous cells. In this case, they did chemotherapy on her, but they did chemotherapy on her bone marrow, because bone marrow is where in the body that the stem cells are that create blood. So you have adult stem cells in your body. These stem cells are cells located in your bone marrow that are responsible for creating your blood cells. Your blood cells don't have a nucleus, so they have a relatively short lifespan, which is why you can go and give blood. How many of you have ever given blood in your lives? Oh man, you guys should go give blood. You're young, healthy people. You might not be able to. They have a minimum weight requirement for giving blood. But you can give blood every 2 months because you drain a portion of your blood out, but then that blood regenerates because there are stem cells that create new blood cells. Your blood cells have this kind of biconcave shape, but they have no nucleus. as a result, they are relatively short-lived. They function for a while and then they die off, they get recycled. And so the chemotherapy that this woman received was chemotherapy that was focused on her bone marrow because that's where the stem cells are that are creating her new blood. Once they killed all of her stem cells, they had to then turn on this gene that produced fetal blood type that doesn't have this problem that sickle cell anemia has. And so once they got rid of her adult stem cells, then they essentially turned on the gene that controls fetal blood production. And so now she is capable of producing fetal blood cells that don't have this defect in their hemoglobin. And so it's one of these cases where they basically used CRISPR to identify a specific part of her genome and turn on the gene that produces a particular type of hemoglobin. This has been used in other organisms to show what can be done with it. These are nematode worms. There's a nematode worm called C. elegans, Caenorhabditis elegans, that people use for genetic work for lots of reasons. But basically, this is a normal phenotype. This is a phenotype called dumpy. The worms tend to get much shorter and fatter than a normal worm is. It's actually called dumpy. It says dumpy or nematodes. And it turns out that that phenotype, that particular growth form of the nematode, is controlled by a gene, and you can induce it using CRISPR to do this. You can change the color of the eyes of fruit flies. You can also change how much tissue is going on in zebrafishes. These are all genetic models. The zebrafish is a genetic model, the fruit fly is a genetic model, the nematode worm is a genetic model. And this is usually where we do these kinds of things first to develop the methods for doing them. And it's actually, the thing about CRISPR is it's a very specific in terms of where you make changes, and it's also very specific the type of change that you make. And this is why people are kind of freaking out about CRISPR. So you can basically take rice and convert it from regular rice to albino rice, once again, using CRISPR in plants. And so people are worried about this because if you've followed any of the news about this, they're worried about designer babies and things like this. There was the controversy last year, this guy in China basically CRISPR'd these two twins, CRISPR'd one of them and didn't CRISPR the other one, and CRISPR'd a twin to basically make them resistant to the virus that causes HIV. And you can do that in China because the controls on what you do with humans are a little more lax in China than they are in the United States. And so it's controversial because if you do this in an embryo, if you do this in a zygote, this one cell that gives rise to all of the cells that become you, that change then becomes a heritable change. It'll be found in every cell in your body, as opposed to the woman who's CRISPR as an adult with the sickle cell anemia case, where only specific cells are being targeted with CRISPR, those cells that are responsible for creating blood. And so this is a controversial technology because it has the ability, if you do it in a very young embryo, of creating changes that will not only affect that individual, but will also then be genetically passed down to later individuals, because the germ cells, the cells in the ovaries and the testes that end up making the gametes, those then get passed on from one generation to the next. And if you do that early enough in the development of an organism, before ovaries and testes have differentiated, have formed, then all of the cells of the body contain those changes, and those changes will be passed on from one generation to the next. Yes?
Are they just worried about how it gets passed on? Like, if it gets passed on bad? Because, being, vaccinated for HIV, that sounds like a really positive thing in general.
So there are a couple of things that people are concerned about at this moment. One is, sometimes when you change something about one aspect of the genome, that has other effects. And so there's this idea in genetics called pleiotropy. We didn't really talk about it, but it's an important concept. It's whenever a single gene controls more than one trait. So we talked about quantitative traits where lots of genes all contribute to a single trait. Those are additive genetic traits. But this is a situation where a single gene can affect multiple traits. And so, so you have genes that turn on growth hormone, and once you reach a certain age, those genes basically stop functioning anymore. You don't have growth hormone being produced, and you stop growing. So you're now, I think, probably at your adult size. You're not going to grow any further. Well, growth hormone controls whether or not in your long bones, at the end of each of your long bones, there's a thing called the epiphysis. It's a section of your long bone that while you're growing, causes your long bones to elongate, and it's cartilage at both ends. And so your bones basically grow. I'm terrible at drawing bones, but there's the epiphyseal plate is here and here, and it grows by producing new bone to either side of this. And so your bone isn't growing in the middle, it's actually growing from the two ends and getting longer and longer. And so long people have epiphyseal plates that were really active, probably because they have differences in growth hormone, because growth hormone affects how active these epiphyseal plates are and how long they stay active. There is a genetic defect in growth hormone regulation such that those genes never turn off, and as a result your epiphyseal plate stays cartilaginous throughout your entire life, and you get longer and longer and longer. These are these extremely gigantic, super tall, thin people who have this genetic defect because it never turns off growth hormone, essentially. Ours turns off, and these cartilaginous plates eventually calcify, and at that point, you're as tall as you're ever going to be. You're as tall as you're ever going to be. You're as tall as you're ever going to be. You're as tall as you're ever going to be. Because you've now lost the ability to grow because your titer of growth hormone has declined. So growth hormone affects not only the length of your long bones, but it also affects all sorts of other things about your phenotype as well. Height is just one of them that it affects. And so one of the potential problems of CRISPR-Cas9 is this was a change to make rice albino, to get rid of the photosynthetic pigment. But what you see is that also had all these other influences on the phenotype. So one of the things that people are concerned about is, okay, you go in and you do this thing in an embryo that's probably good for the embryo. In the process of changing that one thing, have you also then changed a lot of other things that could have problems? It's a little sketchy to do this too soon until you've investigated, okay, if we change the color of the eye of a fruit fly, what other phenotypic effects does it have on that fruit fly? If we make a worm dumpier, does it change that trait, or does it also change all of these other traits? If changing those single traits then has other phenotypic effects, we want to know more about what those other phenotypic effects are before we start doing this in humans generally. In the same way that if you're developing a drug for market, if you're a pharmaceutical company, you do trials on mice and things like that to see if that drug, for example, is going to interact with other drugs that a patient with that condition might also be taking. That's how we get drug interaction information that doctors use when they go to prescribe a new drug for you. The first thing that they will ask you is, what other drugs are you taking? Because they know that if we combine this drug with another drug, that has negative consequences. So it would be nice to know the degree to which changing one trait in an organism will change other traits. And so that's one real health concern about doing this. And I think the other concern that people have is just kind of a fairness concern. If you have the capability to, go in with CRISPR and alter your child's phenotype in ways that make them smarter, taller, more handsome, blonde hair, blue-eyed, whatever it is, you have the opportunity to alter human evolution in a way that we didn't have before. And so there's also that kind of more existential angst that some people feel about doing this, which I get. I'm not sure how I even feel about that at this particular stage in the game. Have you guys ever seen the movie Gattaca? You should watch the movie Gattaca. The movie Gattaca deals with this very directly. It was a movie made before CRISPR was even invented. But Gattaca is about the situation where you can basically pay to have a company put certain traits in your offspring that then make them more successful, such that when you go to apply for a job, they don't actually judge you on what your capabilities are. They just basically, they have what your genotype is for all of the genes in your body, and you're available for certain jobs and not available for other jobs simply because of the genes that your parents put into you. And there's this dude who wants to, go on to an outer colony. He wants to be eligible for being one of the colonists in this new colony in outer space, but they're only making it available to the most genetically superior people. And so he goes through this elaborate process of taking over this other dude's identity. He has to break his bones and lengthen his bones, and he goes through all of this stuff to basically get into this program by basically taking over this other guy's identity. He keeps this guy's crippled, and he basically takes his cells so that he lays the other guy's DNA around places because they go through and they do genetic screens occasionally. It's a weird kind of dystopian science fiction thing. No way that the makers of Gattaca could have foreseen that CRISPR would come along that would potentially allow this kind of thing to happen. So it's actually a more interesting movie to watch now than it was, 15 years ago when it came out, because CRISPR has come along since then. Right now, there's no crops that I know of that have been CRISPR that are available for consumption out there in the world, et cetera, et cetera. In reality, I'm way less concerned about CRISPR being used to turn genes on and turn genes off than I would be about CRISPR being used to move genes from one organism to another. So, for example, if you have BT corn, they've taken a gene out of a bacterium and put it into a corn plant. And in theory, that doesn't do anything to us because it affects insect development, which is very different from our development. And as you'll see in the National Academy of Science report, there's no evidence that there are any negative consequences to that. But I'm always a little more concerned about moving genes from one organism to another organism, in this case from a bacterium to a higher plant, than I would be simply upregulating or downregulating or turning off a gene that already exists in that organism. What CRISPR does is it allows us to have a great deal more control over where exactly we're going into a genome and tinkering with it than, for example, Agrobacterium tumefaciens does. So it's in some ways slightly easier to do and to do more precisely than using a bacterium as a vector for introducing a gene into something. But it's no different in effect. You're taking a gene out of one organism and you're putting it into another organism. In one case, you're doing it in kind of a sloppy, not very controlled way, and in the other way, you're doing it in a very precise way. But it's essentially the same thing, just two different technologies doing the same thing. Questions about genetic engineering? All right, the next topics are all topics that are covered in Chapter 2. You haven't read Chapter 2 because I haven't asked you to read Chapter 2. We're also missing half the class because I did not realize that half the class were soccer players. And Caden has a test that he needs to study for, that I think he's probably studying for right now. All right, we'll pick up Chapter 2 on Monday. Talk about nutrient cycling. Once again, pay special attention to the section about Fritz Haber and nutrient availability to plants and how that changed in the late 1800s, early 1900s.
Do you have the instructions for the presentations and stuff on Moodle?
There's an assignment on there, but I don't have specific instructions. You basically need to be able to explain to the class what the major findings were of your chapter.
Okay.
And so each chapter deals with a different thing. So chapter 3, I've already introduced a little bit of the history of genetic engineering and the process of genetic engineering. So I've kind of covered a certain amount of what's in chapter 3. You guys are responsible for covering chapter 4, 5, and 6. One deals with agronomic aspects, agronomic concerns having to do with genetically engineered crops. One has to do with human health impacts, and one of them has to do with environmental impacts. And so your job with your team is to say, okay, there's this big long chapter where they reviewed all of the science that was available as of 2015 on this topic. What did they find in all of that, and what did they conclude? And there are three kind of ways that they conclude things. They conclude things, 1, there's not a problem, 2, there is a problem, or 3, we might not know enough to know whether there's a problem or not. And so you need to know what those, that's kind of the 3 major breakdowns of the kinds of conclusions that they make. And if there isn't a problem, what's their evidence for saying that there isn't a problem? That means that there's been a sufficient number of studies put towards asking and answering this question, and they've all pretty much turned out a particular way. If there is a problem, the same thing is true there. There have been enough studies done that and they've all turned out in a particular way, saying that this could be a potential problem. But one of the things that you're going to be irritated by, and you need to kind of come to terms with, is that science oftentimes takes a while to get to an answer. And you're going to find repeatedly in the National Academy of Science report saying basically some studies have been done, but not a sufficient number of studies have been done, such that we still don't know the answer to this question yet. It might go one way, it might go another way. For us to conclude that it is one thing versus another thing at this point in time would be premature. It's another one of those situations where scientists really are hemmed in by listening to data. And I think one of the things that students have found frustrating in the past is the National Academy of Science's reluctance to, in some cases, make a conclusion or a recommendation because they just feel that there's not enough evidence there at the time that the report was written, which was 4 years ago, basically. So pay attention to those things as well. You're going to see a surprising, well, I think what for many students is a surprising amount of that, refusing to make a conclusion because they don't feel that the data are there yet. And that's a perfectly scientifically valid thing to do.
Right, yeah.
Don't get upset at that, just, it's like, we need more information. These things are all relatively new technologies. When were you guys born, 1998?
Yeah.
Yeah, these things came on the market just a couple of years before you were born. So this is a change that has happened in your lifetime. Not surprising that we don't have all the answers yet. Okay?
Oh, by the way,
Yeah.
I'll probably send an email just a reminder, but I have a golf tournament in Chicago on Monday. So
Okay, all right.
yeah.
What's the code for Moodle?
The code for Moodle? You haven't been on Moodle yet?
I just realized today I was looking on it.
Pollan, Pollan. It's in an email that I sent you at the very beginning of the semester. Should be easy to remember. It's the author of your book.